Hello. The purpose of this video is just to tie together the two processes that we've talked about this chapter in relation to photosynthesis. We're going to look at the light-dependent reactions and the Calvin cycle and how the two of them work together to generate this entire process. So in order to talk about this, we kind of have to go back to where we started with everything with the chapter. If you remember this diagram from the very beginning of the chapter, I said this is a great little summary of the entire process. And if you know the formula for photosynthesis, it's easy to follow along with where that shows up in the different steps. On the left-hand side of the formula, we have the reactants. We've got 6 carbon dioxide, or CO2, and then our 6 waters, or 6 H2Os. So those are the things that are going into this diagram. The products are then the things that are leaving. So we've got our C6H12O6, that's our glucose, sugar, and then the 6 O2s, our 6 oxygens, that are leaving the other side of the diagram. So if we're looking at this one, it is a very decent representation of just like the general properties of what's happening during this process. The light-dependent reactions, which happen in the thylakoid, absorb sunlight. They then break apart water molecules, creating oxygen, which leaves as a waste product, and ATP and NADPH. These two energy carriers are the things that move on to the Calvin cycle. In the Calvin cycle, ATP and NADPH are used to rearrange the molecules in carbon dioxide, water, and then many of the other things that are already there, like we talked about, things like RUBP and PGA, PGAL, things that are part of the Calvin cycle. So the energy here is used to rearrange those things in order to produce sugar. At the end of that process, the ADP and NADP plus are left over. Those things go back to the light-dependent reactions in order to be recharged again. So we've been over that process many times. What we're going to try to look at today is just sort of representing both of these processes on here at the same time, and then looking at how they are both sort of working to complement each other. Let's try to come up with a nice balance of having both of these things on the screen. There we go. So if we just start walking through this, remember we said that everything starts in photosystem 1 of the light-dependent reactions. Photosystem 1 absorbs light. It uses that light energy to break apart a water molecule. We get 3 things from that water molecule being broken apart. We get hydrogen ions, which stay inside the thylakoid. We get oxygen, which actually leaves and is released as a waste product. Remember, since oxygen is a neutral molecule, it can go directly through the membrane, whereas hydrogen is trapped because it's positive since it's charged. The other thing we're getting is the electron. We said we think of the electron as like what's holding this water molecule together. And once that water molecule is broken apart, that electron's released, it begins moving down electron transport chain. As it gets to the second part of ETC, remember ETC takes a hydrogen from the stroma and moves it into the inner thylakoid. Then the electron continues moving on down to photosystem 1. At photosystem 1, it uses light energy to combine the electron together in order to hold a hydrogen onto NADP+, which makes NADPH. The last step of the process is ATP synthase. This is powered by all of the hydrogens that are on the inside of the thylakoid. They diffuse out through ATP synthase. As they diffuse out, ATP synthase rotates, adding the final phosphate onto ADP in order to make an ATP molecule. So we really end up with 3 products here. We've got oxygen, which is released as a waste product, NADPH, and ATP, both of which are energy carriers. So now, if we go over to the Calvin cycle, and these things would quite literally move to the Calvin cycle. So this NADPH is moving and becoming that NADPH. You know, this ATP is going to wind up being our ATP over here, and then also our ATP on this side. But just to follow things around the Calvin cycle, we said on this side, I think it's easiest to think about the process starting here. We've got RUBP, which is our 6 5-carbon molecule for 30 total carbons. That gets combined together with our 6 carbon dioxide, which are 6 carbons, to give us 36 carbons in PGA. The other thing that happens here is that oxygen is released in this step, since we're not using the oxygen from the carbon dioxide. The problem with PGA is that we can't remove 6 carbons from it. So all this energy is used up. You can see the ATP goes and becomes ADP, the NADPH becomes NADP+, because it's giving up its energy. It's giving up its energy in order to rearrange that 12 3-carbon molecule PGA into PGAl. Once we have PGAl, now those 3 carbons can be removed from it. 6 carbons can be removed from it. These 6 carbons are going to move on in order to become part of our sugar molecule. What we're left with, since we removed 6 carbons from a 12 3-carbon molecule, is 10 3-carbon molecules. So we've got 30 carbons left here. We need to get back to RUBP so the cycle can start all over again. In order to get back to RUBP, this has to be rearranged from 10 groups of 3 into 6 groups of 5, which again requires ATP, requires energy to do that. So once we get back to the beginning here, this whole cycle can happen again. Remember, the Calvin cycle happens continuously. It's light independent, so this is happening in the plant even at night. The final thing to look at then is where some of these products go at the end. Where do the ADP and our NADP plus go? And remember, they're going back to the light-dependent reactions. That becomes this ADP and this NADP plus that eventually get recharged and then return to the Calvin cycle. So if we think about our diagram over here that we started with, the connection between these things, if I can blow this one up, sometimes when I make them really tiny, there we go, they're hard to get. But if we look at this one, the connection between the light-dependent reactions and the Calvin cycle are the energy carriers. So that's really what's connecting the two sides of this cycle together. And that's what I'm trying to stress with you when we're looking at this one, that's really the driving force behind this whole process. The final thing is this guy down here at the bottom. We need to take a look at our 6 carbons that are attached at the end here that used to be attached to PGAl that were removed. In order to get sugar, we're going to add 6 water molecules. And with that, 6 H2O, that's going to give us our C6H12O6, which is glucose. That's the whole purpose of going through this process, is to make these sugar molecules, which are then stored by the plant. So hopefully this was a nice little walkthrough going through those steps. If each of those steps makes sense to you and you understand how these two processes are related to each other, then you're absolutely fine. Then you're in a very, very good position for the test that's coming up, hopefully on Thursday, weather permitting. So we'll certainly see how that goes. But as always, if you have any questions about these things, certainly ask either in class or if you want to come in and practice with the diagrams, either during activity period or at the end of the day or something like that. You're always more than welcome. I think if you can walk me through the diagrams at this point, step by step, then you're certainly more than capable of applying that and answering any of the questions that I'm looking to ask for you on the test. So as always, thank you for watching, and I'll see you in class.